All right, here to complete the next bit, we have looked now at DNA, RNA. We progress now into the idea of a dogma for what biology is built upon, the framework for which all biological knowledge is placed upon. The way we understand today the progression of information from DNA to protein. So again, we call that the central dogma of biology. Understanding that it's not truly a dogma because nothing in biology is incontrovertibly true. A lot of things are challenged every day. Einstein's theory of relativity is currently under analysis. So there's some Portuguese mathematician that claims Einstein made one wrong calculation. So if he can prove that, yes, Einstein was incorrect, all that basis of relativity is, we had to say, well, that's not correct. And we unlearn what we thought was correct. We relearn now the new actual process, right? So that's, again, the idea of how biology differs from a true dogma. So central dogma, quote unquote, of biology. DNA, what we inherit. DNA then undergoes a process called transcription to produce RNA. RNA then is translated to make protein. So we jump ahead again. I believe this is chapter 12. Don't quote me on that. I don't have my book here. It's at the office at EPCC. I can't get into there at the moment. So just look for on the table of contents, look for protein synthesis or central dogma of biology. I think it's titled protein synthesis. So this idea of a progression, right? So we start with one thing, it changes to something else, it changes to something else. And it doesn't technically change, it provides the necessary instruction, the template for the change to happen, right? And I'm gonna elaborate on all the details here, but our discussion will focus on this sort of changing of instruction, right? We're still within the family of nucleic acids, right? So DNA is a nucleic acid, RNA is a nucleic acid. They're just different variations, right? Different sugars, different strands, different bases, but still a nucleic acid. Down here, we're going to have a conversion, a translation from the language of nucleotides to the language of amino acids, right? So again, in white, both nucleotide, both nucleic acid information. In orange, different language of amino acids. So transcription, conversion of DNA to RNA, translation from RNA to protein. And this is, again, what our biology books are built upon, this knowledge. If they come out with some new information tomorrow that, no, this isn't right, well, we gotta get rid of all the books, reprint new books according to the new dogmatic principle that we would have discovered there, yeah? So protein synthesis, right? The formation of proteins. Proteins are anabolized. We undergo anabolic process to connect amino acids together to form proteins. Well, how do we know which amino acids, in which sequence, how many to connect together? Well, that's going to be part of our DNA instruction there. And in this process, again, two processes are required. We call transcription, DNA to the formation of messenger RNA, and then translation, messenger RNA to the construction of amino acids, the primary protein structure. So what we're looking at in this slideshow is this blue straight line to RNA and then the green line to protein. This sort of circular line is possible. DNA can replicate more DNA, but that's for another discussion, that's for another chapter. Right now we're going to look at the transcriptive process and the translative process. So going from a double strand, double helix to a single strand to a convoluted tertiary structure here. So the central dogma of biology, protein synthesis. I'll come back to this slide here in a bit. Let me just start off with the main character in this sort of metabolic process, right? We call this RNA polymerase. RNA polymerase. It's this sort of gray, sort of oblong looking thing. So RNA polymerase is an enzyme, right? And I hope you were able to figure that out, right? Polymerase, ASE. So RNA polymerase is an important enzyme that will do three important aspects, right? It will arrive at a DNA molecule. So in blue, we have our DNA. In blue, we have our DNA. And you notice our 3 to 5 or 3 strand, 5 strand. So this would be 5, 5, 5, 5, 5, 5, 5, 5 to 3. This would be 3, 3, 3, 3, 3 to 5, right? So we're actually going to work on the strand that is going in this direction, 3 to 5, right? So in blue, the DNA strand will be split open, almost like a zipper, right? This is going to be opened up. That's the first role of RNA polymerase. RNA polymerase will open the double helix. And how would we open a double helix? What type of bond will have to be broken? I hope you remember it's the little hydrogen bonds that are connecting the A's with the T's, the C's with the G's, right? So we break those hydrogen bonds, not very difficult to do. And if we break those hydrogen bonds, that strand will separate apart. So after we do that, we then transcribe the DNA. What does that actually mean? Transcription. DNA is not changed. DNA is not altered in any way. But the 3 to 5 strand serves as the template. It serves as a guide. Right? So we keep that strand. And if you notice in blue, we have T, which would correspond with A, which would correspond with T. Well, after the strand is open, it's available now for the transcriptive process. And what transcription is, it works from the 5 to 3 direction. That's why we need the DNA to be going from 3 to 5, so we can be anti-parallel again with the 5 to 3 here. Right? So we're going to construct a new strand of RNA, and we use the template. So if we have a T, well, normally A matches up with T. If we have a G, C matches up with G. So we're not trying to make a double strand. RNA will retain its identity. RNA will be always a single strand. But we use the instructions here to tell us which base to add on the RNA sequence. Now, we have a little bit of a new aspect, right? If I have an adenine on the original DNA, I cannot put T, thymine, on RNA, right? So the DNA has A. We're going to have uracil then, that is sort of a complement of RNA to the adenine base here, right? So we will continue as long as the DNA strand is being opened. As long as we're tearing these bonds, we will continue to construct our RNA polymerase. Our RNA polymerase will continue to transcribe the DNA to form a single strand of RNA. After we're done, we can't just leave the DNA all scattered. That DNA has to then be allowed to close back. So 3 important processes accomplished by RNA polymerase. This enzyme, ACE, A-S-E, makes a polymer of RNA. And so RNA bases, RNA nucleotides being the substrate that work with the enzyme RNA polymerase. So what does it do? Opens DNA, transcribes to form a single strand of RNA, and then closes the DNA back, right? And all this is occurring in the nucleus. So this is going to be the where. We haven't really addressed the where things are happening. We'll start to get to that soon, but this would happen in the nucleus. Yeah? And we will then have started with a double DNA strand, and at the end of transcription, we will have a single mRNA strand. So in this picture, backed up one. So we opened the DNA strand, the strand that was going in the 3 to 5 direction, and we use that as a template, right? Template. So we have an A, we match it up with the U on RNA. C with G, just like normal. A to U, A to U, A to U, C to G, C to G, G to C, A to U, G to C, T with A. Right, so that's that pink process, transcription, which forms our messenger RNA. So different picture, same concept, just different picture of the transcriptive process. So in green, we have our little donut, this green donut looking thing, which represents our RNA polymerase. In blue is our double helical structure of DNA. So we will open our DNA strand. We will initiate our, basically our transcriptive process. I'm not gonna get into all the details here, but we do have to have an instruction that lets us know where to start, and then an instruction to let us know where to end, right? So I'll touch on that briefly, but I'm not gonna get into all of the cellular communication, cellular signals, genetic signals that allow this, but just understand we need an instruction. Which of the millions of A's and T's and C's and G's, which little instruction do we need to transcribe, right? And that's that initiation site. So once we hit that go, right, that initiation, that start, right, we then open, we transcribe, and then we close the DNA behind us, right? That's the role of RNA polymerase, working in the 5 to 3 direction. So it leaves behind then a single green strand of mRNA. It continues working until it's gonna hit that termination signal there. And at the end, after we complete transcription, our DNA is perfectly fine. It's not damaged, not altered in any way. And then now we have our newly formed mRNA sequence. And again, it's not the entire strand of DNA, it's just a short instruction to go, complete whatever needs to be completed in that cell. All right, so I think, again, pretty straightforward. I hope it's not too complex. The main confusion students have with this is how do we address that uracil, right? If we have an A in DNA, we match that up with the U in RNA. And again, that's not the right vocabulary. We're not matching it up. We're just using that template to bring in a uracil to establish our single RNA strand. All right, so after transcription, we go to translation, right? In translation, we're gonna need a couple of things, right? So translation, we're gonna need first our messenger RNA sequence. And we're gonna then analyze our messenger RNA sequence by using codon analysis, right? C-O-D-O-N, codon analysis. A codon is a 3-base sequence on messenger RNA, right? So a 3-base sequence on messenger RNA. So if I have all these bases here, right? And how would I know this is RNA? Because it's only one strand and there's a uracil, right? So maybe with a pen, with a little highlighter, I normally like to underline them. So I'll go 1, 2, 3, underline. 1, 2, 3, underline. Underline 1, 2, 3, underline 1, 2, 3, underline 1, 2, 3. And I designate then the 3-base sequence on mRNA. And we designate those as codons. So if we were to start right here, right? If our mRNA was gonna start right here, CAU would be my first codon. CCA would be my second. And then we start forming our third, AUU. So 3 base sequence on messenger RNA. So another bit of information we're going to need is something called a codon chart, or the universal code chart. I have uploaded one for you here. I think this is a nice, colorful one. You can find others on Google, you know, internet, whatever. In the book, there's one as well. But this one to me is very nice and convenient. And this is basically like a Rosetta Stone, a translation guide. Right? It's 2 languages, right? It's the language of, you know, what does UUU mean? What does the nucleic acid language UUU translate into protein? Well, UUU will translate into phenylalanine. That's a type of amino acid. Over here, CCU translates into proline. So in the blue boxes, we have the nucleic acid information, the codon information. In the tan, light gray, whatever color that is, we have then our amino acids that are coded for by those particular codons. The way we kind of use this chart, we find the first letter on the left. So first letter U, first letter C, first letter A, first letter G. Our second letter we find along the top. So there's a U, C, A, G. And third letter we find along each of these sort of boxes there. So U, C, A, G, U, C, A, G. So if I were to ask you to find, I don't know, let's do UAC. Can we find UAC? If you're given the code on UAC, which amino acid would that code for? UAC. I hope you kind of followed along. You look, okay, well, where's, okay, there's the first letter U. Where does this row intersect with the vertical column here, right? So there's U, there's U, C, A, O, there's U, U and A intersect here. And then again, U, C, A, G. Oh, there's C. So UAC would be the codon that codes for, it's translated into tyrosine. And that's how you would complete the translation process. So again, first we need the DNA strand to know what mRNA sequence we're going to get. Once we have our mRNA sequence, we sort of break it up into different codons. And once we have our codons, we need the help of the codon chart to be able to know which amino acid sequence that we would have. Now, just like in transcription, we needed an initiation, start instruction, and then we need a stop instruction. Same thing in translation. In translation, we need a start instruction. We call it start codon. It's a very, very important sort of instruction, AUG, for the month August, or the name Agustin, right? Augie, that kind of stuff. So if we have AUG, that's the instruction that says, let's go. When the mark gets set, let's start this process. So we'll begin transcription until we reach 3 possible stop instructions. So UAA, UAG, or UGA are 3 ways that we can terminate the translation process. So we're going to keep working, keep working, keep working until we hit one of these, and then translation is stopped. So on your exam, I have given you some questions that I provide you with the DNA. And I don't know if I forgot if I actually designated it, but if it's not there, write it in. the DNA must be going from the 3 to the 5 sort of trajectory there. So if provided the DNA, you then have to construct the mRNA, designate the codons, and then let me know what the amino acid sequence would be. So for example, UGG. UGG, what is UGG? So we come over here. UGG, UGG, ooh, tryptophan, right? Tryptophan, is that what they gave us? Yeah, tell us that's tryptophan, that amino acid there. So from past experience, I see that students don't struggle too much with this. They do sometimes go too fast and they make little either transcription errors or translation errors. So if you make a mistake and you give me the wrong letter here, it's probably gonna give you the incorrect amino acid there. And so just be mindful, you know, work at a pace that enables you to avoid any mistakes during the transcriptive process and the translative process there. All right, so with mRNA, with the codon chart, we still need one other piece of the puzzle, right? During this translation process where we're gonna help the cell generate proteins from mRNA instructions, from codon instructions. So in this picture, we have that mRNA sequence, again, going in the basically 5 to 3 direction. I didn't mention this over here. So under translation, we have the ribosomes. So ribosomes would be kind of like the chefs, right? We talk about you go to a restaurant, you are the DNA, the waiter, the waitress is the RNA, the chef is the one that's actually cooking the food, right? Well, who's actually piecing together the protein? That's going to be the ribosome. Right? That's the chef. So we have the DNA is out there, right? The customers out in the seated in the table. The waiter waitress arrives with their instruction into the kitchen. So the chef now, this brown sort of mushroom looking thing, that ribosome, starts to read each codon by codon, requesting, hey, you, I need you. You, we say codes for phenylalanine, right? So you can imagine a chef, a grouchy chef. Hey, you, we need some phenylalanine over here. Phenylalanine. Well, who's actually going to deliver the phenylalanine? It's a secondary type of RNA that we call tRNA, transfer RNA. So transfer RNA, it reminds me of like the same surgery. You have the surgeon, scalpel, scissors, hemostats, whatever he's needing, yeah, she's needing. So as he requests something, somebody else has to come in here and provide the scissors, the hemostats, whatever the case may be. So we need some phenylalanine right here. Well, tRNA is in charge of bringing the appropriate amino acid to the appropriate location. And again, it's this little cross matching again. We know the codon is UUU, and we have something now called the anticodon, which is going to be the complementary match to that codon. So it's a temporary little match. We know we're in the right location. We drop off the amino acid, and then we go out to get some more scissors, to get some more scalpels, that kind of stuff. So zooming out, we zoom out and we look at the entire process at the same time. And this is where it gets a little bit busy. A lot of things going on, and I do encourage you to look at the video on BioFlix. Look at the notes, follow along with the video. The homework will ask you to do some of this stuff as well. But we will have then our DNA strand in the nucleus. In the nucleus, we have our RNA polymerase. RNA polymerase will then transcribe to form our RNA strand. That RNA strand will be made more concise. Right? No, that's not actually important. Let's cut this out. No, let's make this more simple. That's a run-on sentence. Let's make it, you know, more simple, right? So the message itself gets sort of edited, gets processed. We cap that message on both ends. And then that RNA strand, which is capped on both ends, goes out into the cytoplasm. We're going to come back to these terms, nucleus, cytoplasm. It goes out to the cytoplasm to team up with the ribosome. The ribosome clamps around the mRNA strand. So ribosomes clamping around the mRNA strand. We start reading codon by codon by codon. These little green tRNA molecules bring the appropriate amino acid, and we start to accumulate this primary structure of the protein. They call the polypeptide is a primary structure of that protein. So a lot of stuff going on at the same time. I'm not going to get real elaborate with tRNA. Just know it helps out. On one end, it has the anticodon. On the other end, it will be connected to that amino acid. So it carries an amino acid, associates with mRNA, and also helps work with the ribosomes. All right, so far so good. You think you're a little bit okay with that? Let me see what I can do here. Hopefully I can make this work. Didn't open. So what I'm trying to do, I'm trying to get to one of these files that you have uploaded. Why is it not letting me do this here? There we go. All right, so I hope you can see that. If not, somebody email me, hey, I was not able to see that, but hopefully you can. This is a study guide for your test, and a completed study guide I will give you credit for lab as well. So complete this study guide. It's uploaded already on Pearson. Complete it. It'll give you practice, and it'll give you points for lab as well, right? So number one is just definitions. Be able to understand what these terms are. What is transcription? What is translation? mRNA, codon, tRNA, anticodon, right? And now we're going to get into certain mutations in a bit, yeah? But let's start here. So when we look at this, we look at only a single strand of the DNA. I understand the DNA is double-stranded, but because, you know, trying to make this take up less space and whatever, so I'm only providing you the one strand, the correct strand, and you should remember what direction that strand should be going. So if you're given one DNA strand, which this is how your exam is, I give you one DNA strand. I would like for you to show me the transcribed mRNA and then the correct amino acid chain. So two things you will need, right? You will need to remember that template transcriptive process, and then you're going to need to have that codon chart in front of you. Some things to consider, right? If we look at TAC, well, that's going to code for AUG. AUG, we've heard that before. AUG, Augustine, right? That becomes our start codon, right? So that's one way to double check that you're doing this correctly. If you don't have a start codon to begin with, something's not quite right. So you should have a start at the beginning and a stop at the end. So those are things that you're looking for there. Right? So I'm gonna stop this video here on question number 2. I would like for you to be a little bit confident with that, get some practice with this, and then we're gonna continue with the next slideshow, looking at mutations. Right? Mutations make sense only if we understand the non-mutations, how things are supposed to work first. Right? So let's get very confident with transcription, translation, and then we'll complicate things with mutations. So with that, let me get this thing converted and started to upload, and keep an eye out for the assignment, the protein synthesis assignment. Bioflix videos on Pearson are very useful as well. It shows everything happening at the same time, a little like a computer animated picture video makes a lot more sense than just static images there, right? So goodbye for now, and we'll continue with the next discussion a little bit later.